Good afternoon. I'm Lynn Markey, Director of the Coalition for Life Sciences, and I'd like to welcome you to the 20th season of the Congressional Biomedical Research Caucus presentation. I'd like to thank our co-chairs of the caucus, Representative Brian Bilbray from California, Mike Castle from Delaware, Jackie Spear from California, and Rush Holt from New Jersey. Without their commitment and dedication, we would not be able to continue with the caucuses and the amazing briefings we're able to present to you. I'd also like to thank the Howard Hughes Medical Institute for its support of the caucuses. They have provided us with a generous grant and truly allow for these briefings to be held with lunch and travel support for our speakers. So we're very grateful for that. I also want to point out, as you can see Tony in the back, we do videotape these briefings. You can find past briefings on the CLS website at www.coalitionforlifesciences.org. Register for our RSS feed and be alerted for future postings. So today I'd like to introduce to you Dr. Daniel Silver, and he is at the Dana-Farber Cancer Institute in Boston, Massachusetts. Dr. Silver was an undergraduate at Harvard College and received his MD from the University of California in San Francisco. He is currently an assistant professor of medicine at the Dana-Farber Cancer Institute. He sees patients with breast cancer as well as those at high risk for breast cancer and runs a research laboratory. His research interests include DNA repair and tumor, I haven't had a chance to read this, so pardon me. Tumor genesis.
Close, tumor genesis.
Sorry. As well as more applied questions in breast cancer. He's the author of numerous scholarly publications, which range in scope from the basic molecular biology of cancer to clinical trials. He is also an enthusiastic teacher of medical students, interns, residents, fellows, and postdocs. So please join me in welcoming Dr. Silver to the caucus.
Thank you very much. Thank you very much. I'd like to thank the Congressional Biomedical Research Caucus, the Coalition for the Life Sciences, Lynn Markey, Keith Yamamoto, and a personal note of thanks to Dr. Michael Bishop, who's been involved with the Coalition for the Life Sciences from the beginning and who organizes this series. He actually was one of my earliest mentors in science, and almost 30 years ago, he heard my first public talk in science. And I gotta say, it wasn't very good. In fact, it was a lot worse than not very good. And so I'm thankful and grateful for the fact that he has more confidence in me now, some 28 years later, than he did then. Though I think those of you who might know Dr. Bishop might realize that he's not in the room, so he may be hedging his bets, and you could be in for a rough ride. So the subject today is PARP inhibitors, which are a new drug therapy for cancer. And what I'd like to do is give you a quick introduction to tumor development, tumorigenesis, so that we all start at the same place. And I'll emphasize the central role of DNA repair defects in tumorigenesis and talk to you about therapeutic opportunities and difficulties in cancer drug development. I'll then give you DNA damage and repair 101, which is necessary so that you can understand how this new class of drugs, the PARP inhibitors, take advantage of defective DNA repair in certain tumors. I'll then give you some examples of clinical applications of PARP inhibitors, and then talk about the problem of resistance and challenges facing cancer therapy in general. And then at the very end, a few thoughts about the public perception of progress in cancer research. So oncogenes and tumor suppressors are the stuff of cancer development. Most of you know that oncogenes and tumor suppressors are mutated during the development of cancer. During the process, it takes a normal cell, which doesn't go very fast, responds to its environments appropriately. And once mutations in oncogenes and tumor suppressors start to occur in a given cell, it turns into this, which divides very rapidly, doesn't care much about the environment, and goes on to cause great harm to the host. Now, oncogenes are genes that encode proteins that have growth-promoting function. They're like the accelerator in that car. And when oncogenes are mutated during the development of a tumor, oncogenes get stuck in the on position, just like the accelerator in certain cars. Tumor suppressor genes, on the other hand, are the opposite. They are genes that either directly or indirectly have growth restraining function. They're like the brake. And when tumor suppressor genes are altered during the development of a cancer cell, they get inactivated by mutation. So this is kind of a summary of our current understanding of how cancers develop. Normal cells undergo DNA damage all the time. Many people don't realize that most of that DNA damage is from endogenous processes. It's part of the normal life of a cell. There are estimated to be about 5,000 separate occurrences of oxidative DNA damage during 24 hours in every one of your cells. And there are also occasionally exogenous chemicals and so on that also cause DNA damage. The cell has evolved many pathways to repair this damage, and they are very efficient. And almost always, the DNA damage is repaired without mutation, restoring the normal cellular state. Every so often, there's a failure of DNA repair, and this leads to the accumulation of mutations. And if a given cell starts to accumulate mutations that activate these oncogenes, inactivate tumor suppressor genes, and other alterations that are necessary to take place, eventually cancer can ensue. So if you're in the business of wanting to make drugs that target cancer cells but spare normal cells, the logical place to look is genes that are different in the cancer cells compared to the normal cells. So the obvious place to start looking to develop drugs are drugs that target oncogenes and tumor suppressors. Now, in principle, oncogenes are easier than tumor suppressors to target with drugs. This is because drugs tend to be small molecules that bind to proteins like a lock and key and gum up the works of those proteins and prevent them from doing their normal thing. So oncogenes are added functions in tumors. So as an added function, it's relatively easy, though my pharmacological colleagues will kill me for saying this, it's relatively easy to inhibit an extra activity, though some targets are clearly easier to inhibit than others. Tumor suppressors, on the other hand, are missing functions in tumors. And therefore, because they're not there, it's very hard to make a drug that adds a complicated function, whatever that function of the tumor suppressor may be, to all cells within a tumor. So how do you target the untargetable? That is, those oncogenes that are difficult to target pharmacologically, or those tumor suppressors that are gone in tumors? Well, there's a concept from classical genetics, synthetic lethality, that offers a way forward in these situations. This is a term from classical genetics that describes the relationship between two genes. Two genes are said to be synthetic lethal if mutations in each alone does not kill the organism, but mutations in both together in the same organism cause death. So this little table summarizes this situation. If you have gene X, say in a yeast, and gene Y in that same yeast, if they're both not mutated and normal, there is no effect, obviously. If gene X is inactivated by mutation, but gene Y is still present, the organism is perfectly fine. And the reciprocal relationship also occurs. If gene Y happens to be mutated, but gene X is normal, the organism is still fine. However, if both gene X and gene Y are mutated, death ensues. Let me give you a couple of examples. Suppose a yeast organism requires a particular amino acid for survival, and it has two separate pathways that are responsible for making this amino acid. And the pathways are completely independent. So if there's a mutation that inactivates one of these two pathways, the yeast can still make the amino acid with the other pathway, and so the yeast survives. And also the other way around, if this pathway is mutated, this pathway is still present, and the yeast survives. But if there are mutations that inactivate both pathways, the yeast can't make this amino acid that it needs to grow, and it dies. A second example comes from actually DNA repair. Polymerase, which is the enzyme that copies DNA in preparation for cell division, has evolved to be very, very good at this and make very, very few errors. And one of the ways it actually prevents errors from occurring is it's always looking behind it, looking at the DNA that it's just replicated to make sure that it's a perfect match to the template. And if it senses that it's just added a base that is not the right base, it actually shifts gears, turns around, goes back, and re-synthesizes that strand to fix that problem. That's called proofreading. There's a totally separate repair pathway called mismatch repair. This is not linked to DNA replication. And this repair system floats around in the cell looking at the DNA all the time for bases that are not matched, where an A is not opposite a T or G is not opposite a C. And so this is always operating in yeast. It turns out that a yeast organism can tolerate the deletion of proofreading capacity as long as it has mismatch repair. And likewise, it can tolerate the loss of mismatch repair as long as it has proofreading. though lesions in either one of these two systems alone does cause a somewhat increased mutation rate. However, if the yeast loses both proofreading capability and mismatch repair, the number of mutations that accumulate is unacceptable and the organism dies. So therefore, proofreading capability and mismatch repair are so-called synthetic lethal. Now, Leland Hartwell, a very distinguished American geneticist who also is a Nobel Prize winner, and Stephen Friend, a very distinguished cancer biologist, wrote a very influential review article in 1997 they published in Science that pointed out that that synthetic lethality could be used to screen for new targets for drugs and tumors. Let me tell you how that might work. Suppose gene X is actually a tumor suppressor gene. So I've told you that tumor suppressor genes can be mutated in a cell, and they don't cause death necessarily of that cell. And there are changes that occur on the way to developing a cancer. What you would do is look for another gene, gene Y, which mutations by itself in gene Y don't cause any harm to the cell. But mutations that are in the same cell as having that inactivated tumor suppressor are not tolerated, that cause death. If you can find gene Y that causes death only when that tumor suppressor, gene X, is mutated, then gene Y could then be a target for pharmacological development of anti-cancer drugs. And my colleague Bill Kalin at the Dana-Farber Cancer Institute updated these ideas in a review 5 years ago, in which he incorporated newer ways to inhibit genes, shRNA and siRNA, into this general idea. So these are the two gentlemen that ushered in the modern era of biology, Watson and Crick, standing in front of their first model of DNA. They published a paper in Nature in 1953 that changed absolutely everything about biology. The paper is only 12 paragraphs long, unbelievably laconic. And they don't really talk much about the implications except for this classic understatement in the last paragraph of the paper. They say, "It has not escaped our notice that the specific pairing we have postulated, that's A's and T's and G's and C's, immediately suggest a possible copying mechanism for the genetic material." Obviously one of the fundamental puzzles prior to the discovery of the structure of DNA. What they didn't say, but which was no doubt completely apparent to them and any biochemically minded biologist reading their initial paper, is that the molecule of heredity, DNA, is in fact a chemical molecule. And chemical molecules are subject to chemical damage. And so chemical damage to DNA results in mutations. And the problem of how the organism avoids accumulating mutations was obviously brought to the fore by this structure. And this engendered more than 50 years of research into how DNA damage is repaired in organisms. The types of DNA damage are actually pretty obvious. The DNA chain can be broken, either single-stranded breaks or double-stranded breaks, and the DNA chain can rust. Rust is just oxidation. And those are the two main kinds of DNA damage that accumulate in cells that the cell has to deal with. Now, the cell has multiple repair pathways that I've outlined here. These are the main ones. There are others. To deal with this, these multiple repair pathways are all independent, and many of them are redundant. So a given lesion in the DNA can be dealt with by any of these, potentially any of these repair pathways. Direct repair is used by some organisms to fix UV damage. So UV damage actually causes cross-links between adjacent bases in the DNA. And some organisms have enzymes called photolyases that directly repair that damage. Base excision repair, which is important to our story later on, is a process that recognizes oxidative DNA damage, that is, oxidized bases. And what it does is it cleaves out that oxidized base from the sugar backbone, cleaves the sugar backbone, removes the sugar backbone along with the adjacent bases for 2 or 3 or 4 bases on either side of the lesion, and then catalyzes a normal replacement, such that the DNA looks just like it did before the damage took place. Nucleotide excision repair works slightly differently. This is a process that recognizes bulky DNA adducts that have been added to bases, large modifications, chemical modifications of bases. And what it does is it actually removes about 50 or 100 bases along with the sugar backbone on either side of the lesion, leaving the other strand of DNA intact, and then it polymerizes a new replacement to return the DNA to its pre-damaged state. Homologous recombination is a process that uses a template other than the damaged strand of DNA to fix an area of damage. And the way this works is, suppose this is an area of DNA after DNA replication, but before cell division. So after DNA replication, but before cell division, every strand of DNA is copied so that every strand has an exact copy of itself, so that there are now two duplexes prior to cell division, and each of the resulting cells after division will receive one of these duplexes. Suppose that one of these duplexes undergoes some damage that results in a double-stranded DNA break. Homologous recombination realizes that there is a template that is undamaged nearby. It uses the undamaged template to polymerize past the area of damage, and then comes back to the original piece of DNA, making this complicated structure. And this complicated structure is then resolved in such a way that you have two completely normal, unmutated strands ready to go for cell division thereafter. Now, I've mentioned to you that these processes are partially redundant, that the same kind of DNA damage can be fixed by any of these processes in certain instances. The situation is even a little more complicated in that DNA replication can interconvert certain kinds of DNA damage. So what I show here is one double-stranded helix being replicated prior to cell division. And the polymerase that's replicating this red strand and creating this new blue strand has run into a modified base, which has a bulky adduct, a bulky chemical adduct that's been added to it during the course of normal cellular metabolism, for example. So the polymerase gets stuck here. It can't copy this base anymore, and it just stalls. So what happens is, after this region thereafter becomes single-stranded. So you have what's called a single-stranded nick or a gap. Single-stranded nicks and gaps are actually relatively unstable, and they can break, so that you can end up with a double-stranded break. So I've shown you then that replication can take a chemical modification of DNA and turn it into a double-stranded break, a different kind of DNA damage. So not only are these partially redundant, but DNA damage can kind of interconvert, requiring the participation of different DNA repair pathways. Important to our story today are BRCA1 and BRCA2. They are tumor suppressor genes. They were cloned 15 and 16 years ago. They were isolated because there were families that had, very tragic families, that had remarkably high numbers of early onset breast and ovarian cancers. These are families where individuals in their 20s, 30s, and 40s were being diagnosed with breast cancer, and often there were families where sisters and mothers or sisters and grandmothers were affected by this syndrome. And by tracking a region of DNA that was associated with disease, these genes were isolated. The way they work is that people who are prone to early onset breast and ovarian cancer are born with one good copy of BRCA2, for example, and one bad copy of BRCA2. This situation is fine, and in general, cells that look like this have completely normal BRCA1 and BRCA2 function. The problem is that rarely a cell loses the remaining normal copy. And if you lose the remaining normal copy, then you have no function. And tumors then develop in this situation. People who are born with this situation, being heterozygous, cancers arising in those individuals account for about 5% of breast cancer in the United States and 10% of ovarian cancer in the United States. And it turns out that both genes are required for homologous recombination. So going back to our scheme as to how cancers form, the way in which BRCA1 and BRCA2 function as tumor suppressors is not directly by inhibiting growth. What they do is, in the absence of either one of these genes, there is a failure of DNA repair. And so rather than efficiently fixing DNA damage, more DNA damage gets turned into mutations, accelerating the activation of oncogenes and accelerating the inactivation of tumor suppressors and other cancers. So cancer occurs more frequently and earlier. So in this situation, there may be actually opportunities for therapeutic opportunities in the following way. I've told you that cells that are missing BRCA1 or BRCA2 cannot perform homologous recombination. And I've also told you that the DNA repair pathways are partially redundant. So perhaps a cell which has lost homologous recombination is more dependent on other repair processes to fix DNA damage. And that turns out to be the case, and it turns out that base excision repair, cells that are missing homologous recombination are particularly reliant on base excision repair and vice versa. So base excision repair is a process that involves an enzyme called poly-ADP-Ribose polymerase. And pharmaceutical companies are very fond of enzymes because they're pretty good at creating drugs that inhibit enzymes. So what this enzyme does is it modifies proteins by adding branched chains of a particular chemical called poly-ADP-Ribose. And the way it does this in base excision repair is PARP1, this enzyme, is attracted to the site of DNA damage, and it begins to add these branched poly-ADP-Ribose chains to all the proteins that are in the vicinity of this break. What that modification does is that it attracts all of the activities required for base excision repair to that locus and sets up the whole process of base excision repair in motion. So in the absence of PARP1, or if this enzymatic activity of PARP1 is inhibited, base excision repair no longer occurs. Two groups in 2005 published published papers back-to-back in Nature, which pointed out that cells that are defective in BRCA1 or BRCA2 are specifically susceptible to inhibition of PARP1. In other words, mutations in BRCA1 and PARP1 are synthetic lethal, or mutations in BRCA2 and PARP1 are synthetic lethal. The way they came about this is they noticed that cells that are missing PARP activity are particularly dependent on homologous recombination. This is from one of these two papers. And what it shows is this is the nucleus of a cell where the DNA is stained blue, and a particular protein called RAD51 is stained red. When the cell is unperturbed in its normal state, there's very little red staining. But when you add a PARP inhibitor that prevents PARP from functioning, and therefore it prevents base excision repair from functioning, the cell accumulates lots of these little red dots. Now, RAD51 is a key protein for homologous recombination. It's the activity that finds the homologous sequence in the template that's going to be used to repair a particular piece of damage. So this suggested to these authors that in the absence of base excision repair, homologous recombination was called into action to take up the slack, to fix all the mutations that base excision repair normally was going to fix. This is some data from one of those two papers in which three chemicals, two PARP inhibitors and a related chemical that is not a PARP inhibitor, were used in a very specific cellular setting. This plots survival of cells versus increasing amount of each of these chemicals. And it compares BRCA1 deficient cells with identical cells that are identical except that these are BRCA1 normal, these are BRCA1 deficient. And again, BRCA1 deficient, BRCA1 normal. These are BRCA2 deficient, BRCA2 normal, and so on. So what you can see is that the cells that are BRCA1 or BRCA2 deficient are much, much more susceptible to the PARP inhibitor than are normal cells. In fact, those of you in the cheap seats can't read this, but this is actually 3 orders of magnitude different. That cells that are BRCA1 deficient or BRCA2 deficient are 1,000-fold more sensitive to these drugs than normal cells. This is a very defined research situation. These are actually mouse ES cells. They're otherwise normal cells. Cancer cells are a little more messy, but this still works in cancer cells. This is an experiment I performed some years ago in which I took a BRCA1 deficient breast cancer cell line that grows in culture, and I added back BRCA1, and I added increasing concentrations of a PARP inhibitor and plotted the surviving fraction. And what you can see is the addition of BRCA1 increases resistance to this PARP inhibitor. I put this here to show you that it's not only BRCA1 cells, tumor cells, that are potentially therapeutically targeted by PARP inhibitors. This is a cell line that is not deficient in BRCA1 and BRCA2, but it is a breast cancer cell line. And presumably this cell line has a mutation somewhere else in homologous recombination that makes it just as sensitive to the PARP inhibitor as the BRCA1 deficient cell. So To recap what I've said, normal cells have base excision repair, which is dependent on PARP1, and they have homologous recombination, which is dependent on BRCA1 and 2, and they repair their DNA just fine. Cells that have a BRCA1 mutation, say the tumor cell in somebody who was born heterozygous for BRCA1, that tumor cell can survive because it has other DNA repair pathways. It does accumulate mutations more quickly, but it doesn't have an overwhelming number of mutations because it has other DNA repair pathways, including base excision repair, present to help maintain an acceptable level of mutagenesis. Normal cells, say the non-tumor cells in somebody who is a heterozygote, because one copy of BRCA1 is perfectly fine to give you normal homologous recombination, they don't die if you inhibit PARP with a cancer drug, because they have homologous recombination to take up the slack of the increasing mutations that are not corrected by base excision repair. But the tumor cells, which are completely lacking BRCA1 and are exposed to drug, die because this creates a situation where there are unacceptably high numbers of mutations and DNA damage that cannot be repaired. So what's the big deal? Why is this all so good? So PARP inhibitors target a defect, the loss of normal DNA repair that's present only in tumors, not in normal cells. So since normal cells don't share this defect, these drugs are relatively non-toxic. Many kinds of cancers have defective homologous recombination, and it's currently a hot area of research to try to figure out which cancers have defective homologous recombination and can be targeted in this manner. Many PARP inhibitors are oral and convenient. They can be used by themselves as monotherapy. They also may synergize with many kinds of chemotherapy. And when you add a PARP inhibitor to many kinds of chemotherapy, it turns out that many conventional chemotherapy agents are mutagens. They modify DNA and they create DNA damage. And you can imagine by inhibiting DNA repair in that context, you might broaden the number of the kinds of cancers that might be targeted by a PARP inhibitor plus chemotherapy. It's a new target, that is defective DNA repair, for modern targeted cancer drug development. I say, well, kind of, because a lot of the chemotherapy that in the bad old days, the standard chemotherapy drugs actually do target cells with defective DNA repair, but in a relatively blunt, non-surgical way, just by damaging DNA. And lastly, they're the first drugs developed with this concept of synthetic lethality firmly in mind. And no doubt there are many more cancer drugs to be developed with this idea firmly in mind. So it turned out that this was a relatively felicitous situation in terms of drug development, because multiple drug companies were in the business of developing PARP inhibitors even before we knew about synthetic lethality with homologous recombination. This is because there was some suspicion that PARP may be involved in other processes that might be useful to inhibit pharmacologically. In fact, there was an idea that PARP1 may be involved in cellular damage after reperfusion. So after a heart attack is fixed or after a stroke is relieved, PARP inhibition may help limit the amount of tissue damage. And that's remaining to be tested. But because of that idea, and because of the idea that PARP inhibitors may synergize with conventional chemotherapy, multiple pharmaceutical companies were in the game of making PARP inhibitors. For that reason, after these papers were published in 2005, it was possible to start conducting clinical trials in relatively short order. This is in fact the first clinical trial using PARP inhibitors that was published. This was published in the New England Journal of Medicine last summer, and I show you a subset of this trial, focusing on the use of this PARP inhibitor as monotherapy by itself in patients with ovarian cancer who are heavily pretreated, who had BRCA1 or BRCA2 mutations. This is a pill taken twice a day, and these patients, 35 of these patients volunteered for this trial. 19, or 54% of the patients who were in this trial, showed tumor shrinkage, and the average duration of response was 24 weeks, 6 months. So one pill twice a day, on average, held these cancers at bay, and some of these patients had a response that was longer than a year. This is another trial which has become public at the American Society of Clinical American Society of Clinical Oncology last summer. This is a trial that adds a PARP inhibitor to chemotherapy. The patients enrolled in this trial had disseminated triple-negative breast cancer. This is a particularly aggressive form of breast cancer, and they had metastatic disease. They were all quite sick. And here, the PARP inhibitor, which is from a company called Bipar, which has since been bought by Sanofi Aventis, was combined with conventional chemotherapy, and patients were randomized to this triple therapy versus just the conventional chemotherapy alone. Patients who had the triple therapy, that is the chemotherapy plus the PARP inhibitor, had stabilization or shrinkage of their cancers on average about 3.5 or 4 months longer than patients who are on the conventional chemotherapy alone. Quite surprising, in this kind of an exploratory trial, there was actually a survival advantage. Patients who had the PARP inhibitor plus chemotherapy lived an average of about 3 months longer than the patients who had the conventional chemotherapy alone. A very striking result, in a therapy that's being in the early stages of development. Let me give you a clinical anecdote that shows what these drugs can mean to an individual patient. This is a patient of one of my colleagues at the Dana-Farber Cancer Institute, Dr. Ursula Matawanis, who's an ovarian cancer expert. This is a patient in her 40s who presented in 2001 with abdominal swelling and pain. A CT scan showed a diffuse tumor, and at surgery she was found to have an advanced ovarian cancer. At the same time, she was also found to have a small breast cancer. Obviously not a good turn of events, but this is a clinically much less difficult problem than her ovarian cancer. This was not a coincidence. She was found to have a BRCA1 mutation, and as I've told you, patients who have BRCA1 mutations are at much higher risk for both breast cancer and ovarian cancer. This is a way of plotting her clinical course. This is a blood test for CA125. This is a protein not normally found in blood. And its level in blood reflects the amount of ovarian cancer that is growing in her body. So when she first presented, prior to her first surgery, she had a very high level of CA125. Surgery brought it right down. She then received chemotherapy with conventional chemotherapy, and this was enough to keep her cancer regressed and at bay for 2 years. Unfortunately, after 2 years after her presentation, the cancer grew back, CA125 rose. She had surgery again to remove as much cancer as possible, and she had another round of conventional chemotherapy with carboplatin and Taxol. 2 years later, the tumor recurred, and she had more conventional chemotherapy. And unfortunately, this is a very common situation with ovarian cancer, where it keeps coming back, and each time it comes back, it's harder to treat. After this last round of chemotherapy, she developed an isolated single liver metastasis, and this was treated with a physical means of destroying the tumor, radiofrequency ablation. In 2007, the tumor came back yet again, and she was enrolled in that clinical trial, the very first one I described to you that was published in the New England Journal of Medicine, with the drug olaparib. Again, to remind you, these are pills that are taken twice a day. And when she began to take these pills, her CA125 promptly fell to normal levels. And for 16 months, her cancer was held in remission by 2 pills a day. These are pills that are very well tolerated, a little bit of GI upset in some patients, but really not too bad. Some fatigue in some patients, not too bad. So for 16 months, she was not in the infusion room. Her hair grew back. She wasn't nauseous. She didn't have to come into the cancer center for infusions of chemotherapy every week, and she was able to enjoy her family life, not feeling so much like a cancer patient. Unfortunately, after 16 months, the tumor recurred. She's since been on 2 phase 1 trials and is still with us. So why is it that tumors become resistant to PARP inhibitors? This is metastatic breast cancer. And I've told you that many tumors have lesions in DNA repair. So that means that these tumors are acquiring mutations all the time. And it means that this cancer cell may have a small number of mutations that are different from this cancer cell, that are different from this cancer cell, that are different from that cancer cell. So even before we treat patients who have disseminated disease, there is a large collection of mutations that some of which potentially encode resistance. And it turns out that that's exactly the case. So I've told you that BRCA1 or BRCA2 loss greatly accelerates the development of cancer. and they are tumor suppressor genes in that manner. But if you think about it, once you make the cancer, the continued loss of BRCA1 or BRCA2 is no longer necessary. Once you've accumulated enough mutations in oncogenes and enough mutations in inactivated tumor suppressor genes and a few other mutations, you have the cancer and you no longer need BRCA1 or BRCA2 loss. So if a cell or two within the tumor mass has reverted back to normal BRCA1 or BRCA2, the PARP inhibitor will kill all the other tumor cells, but that remaining tumor cell that's reverted BRCA1 and BRCA2 will continue to grow and eventually come back and cause a relapse of the cancer. This, in fact, does happen. This is a study by Taniguchi and his colleagues that was reported in November of last year. But this is not a study of PARP inhibitors. This is a study of a conventional chemotherapeutic drug, platinum. Platinum works by targeting cells that have defective homologous recombination, and it is an effective conventional chemotherapeutic drug for patients who have BRCA1 or BRCA2 mutations. So this looked at 18 patients who had BRCA1 or BRCA2 related ovarian cancer. And in 18 cases that were platinum resistant after platinum therapy, 10 of these had mutations that reverted BRCA1 or BRCA2 back to normal. It turns out that 13 of 15 that were studied carefully had mutations that restored the normal sequence of BRCA1 or BRCA2, and 2 had new frame shifts. What does this mean? It turns out that the DNA code is very much like letters in printed language. So patients who are born with BRCA1 or BRCA2 mutations in every cell in their body often have what's called frame shift mutations. That is, a deletion of some odd number of letters such that everything in the code that comes after that deletion is nonsense. Okay, so all I've done is shift 2 letters over. Okay? Now, in those tumor cells that are resistant to the therapy, that grow back and cause a relapse, what's happened is, though they started like this, at random, the same 2 letters are inserted exactly where they should have been to start with and have created a normal wild-type BRCA1 or BRCA2. So 13 of 15 of those patients had that kind of a change. Even more incredible is what's called second-site reversion. Again, the same kind of mutation, causing things to be out of frame. And in a second-site reversion, 2 random letters are inserted somewhere else, such that you have normal sequence going to the original mutation, a bit of gibberish, 2 random bases inserted, or letters in this case inserted, such that the frame goes back to normal. And if this is close enough to the original mutation, the protein that's formed, though it has some change from the normal sequence, is close enough to function. And this happens as well. So what I'm telling you then is that patients who have disseminated cancers, particularly those cancers with DNA repair defects, but many cancers in general, have a great deal of genetic diversity. And the real enemy here is that cells are very small. So 1 cubic centimeter of tumor, which is a thimbleful, contains 1 billion tumor cells. Now, I know you guys are used to dealing with billions around here, but in fact, 1 billion is a very powerful number. And so even if the mutation rate is rather low, most metastatic patients who have metastatic cancer that is spread throughout their bodies has much more than a thimbleful of DNA, have much more than a thimbleful. So they have billions and billions of tumor cells that are making mutations all the time, even before they're treated. So this leads to the rapid accumulation of many, many mutations even before therapy. Therefore, many mutations exist encoding therapy resistance before therapy is even started. And what I've shown you is one consequence. So in summary, the PARP inhibitors are a new drug treatment primarily for certain tumors with defective homologous recombination. Their development represents one payoff of years of basic research into both DNA damage repair and classical genetics. They are relatively non-toxic compared with chemotherapy, and they may be beneficial both given by themselves and also combined with chemotherapy. They take advantage of this concept from classical genetics, synthetic lethality, to target an aspect of tumor biology, defective homologous recombination, which would otherwise be very difficult or impossible to drug directly. And they represent the first fruits of the concept of synthetic lethality applied to cancer therapy, and hopefully there'll be many more drugs to come based on this concept. And I've also shown you how the development of resistance will be an issue for many patients and points out one of the barriers to curing metastatic cancer. A few words about the public perception of all of this. These are two quotes from the American television media when that New England Journal of Medicine paper in 2009 was published, that was the first PARP inhibitor trial. These were on the major US television networks. One commentator said, "Researchers in Britain say they have found the holy grail of cancer research." Now, I'm not so sure that the researchers actually used those words. This is an interpretation by the newscaster. But, you know, I think this is all very good. This is all very clever research. It's all very smart application of basic science to the benefit of patients. But I really don't think that this is the holy grail of cancer research. Another commentator said, "Just released research about a new class of drugs called PARP inhibitors is the most exciting development in cancer research in a decade or more. In just a few years, it could save thousands of lives." Well, that may be true, but let's wait for in just a few years to see how many lives it really does save. Again, lots of things have happened in the last decade of cancer research that we can be happy about, and this is just one of them. This kind of media hype is really widespread. Here, for example, is, in my mind, what was the granddaddy of them all. Some of you may remember this was the cover of Time magazine in 1980. There was a very large article. This came out right after interferon was cloned. This was in the heyday of molecular cloning. And the article promised that interferon would make a major, major dent in cancer therapy. Now, most of you know that never came to pass. Interferons are useful in certain realms of medicine and certain cancers, but a very small number of them. It took me 5 minutes to find all of this on the web. So the public is constantly bombarded with this kind of stuff all the time. And I think that this contributes to public frustration with cancer research. I think that expectations are fueled by media reports such as the ones I've shown you. And to be fair, you know, a number of these media reports are the fault of my colleagues and myself. We work very hard on these basic and clinical problems for years, sometimes 5 years or a decade, and we come out of the lab or out of our offices blinking into a reporter's microphone, and you know, my colleagues are justifiably proud of what they do. And many of my colleagues don't have a real complete picture of how difficult it is to take a basic science breakthrough and put it into the clinic. They're basic scientists who've been working in the lab, and to them the hard part is the basic science, and they don't really have an overview of how difficult it is to translate that to the clinic. And vice versa, my clinical colleagues don't necessarily have a very good idea of the basic science involved with some of the things that they are doing. And so sometimes unmeasured statements are made by people in biomedical research that lead to these kinds of articles. On the other hand, I think there may be one or two of you in this audience who have had experience with reporters that tend to overstate things. And this is a problem as well. That reporters, of course, want to write articles that look important. And so this is really, oops, sorry about that. This has really been a situation where I think the public frustration with cancer research has become palpable because they're reading this all the time, but yet, and you know, their Aunt Anne still has ovarian cancer and is doing very poorly, or their cousin Billy just died of lung cancer, and how come all of this breakthrough stuff hasn't helped them? In many ways, progress in cancer therapy lags behind other areas of medicine, for example, infectious disease. And we've come to take medical effectiveness for granted in other areas of medicine, so we wonder why not cancer. And I think the public also doesn't really have an understanding of the difficulty of the cancer problem. It's really hard. And lastly, tangible progress, as I've described, for example, in the PARP inhibitors, is often incremental, and sometimes it's pretty arcane. And I think you'll agree that the PARP inhibitors need a little explaining for people to understand. So I think the American public has come to think of cancer research like they have the same feelings they have as if they were in this traffic jam with this sign. That, you know, this all sounds great, but it really hasn't helped anyone. But, you know, I hope I've convinced you that we are making progress in cancer. The progress is incremental, but it's steady. And I think our progress in cancer is going to increase greatly in the next few years with the advent of high throughput sequencing and high throughput techniques where we can really start to profile individual tumors. And though I don't think there will be a holy grail that's discovered, I think that our progress will be greatly accelerated, and hopefully we'll make progress in this terrible disease rather rapidly. Thanks a lot for your attention.
So I understand your concern about news stories that probably overpromise. But given that the American public funds a great deal of this research, what should they be told about research?
Oh, I think there are ways of reporting breakthroughs that are straightforward and really say what the breakthroughs are. Even when you use the word breakthrough, it has certain connotations, but words that describe research accomplishments. So, PARP inhibitors, the title can be something like, PARP inhibitors represent a new way to develop, are the first drugs of a new wave of pharmacological development, something like that. Interferons are a class of drugs that may help cancer patients, because when that cover was published, we had no idea that they were going to affect cancer therapy. So I think that many of my colleagues are really careful about what they say to the media, and I don't mean to say that it's all our fault. But sometimes it is, and there are good reporters out there too. But despite all of that, sometimes this stuff happens.
Yeah, I was a little confused about the when the BRCA gene mutates back to normal, because then doesn't it just become a tumor suppressor gene?
Yes, but the damage is done. So the way in which BRCA1 and BRCA2 cause tumors is they accelerate the accumulation of mutations. And once those mutations occur, they're part of that cell forever. So once you've accumulated enough mutations to cause the cancer to occur, their continued loss is no longer required. So that's why I kind of call it the Achilles heel of the Achilles heel.